Hello, this is Rupinder Syal and welcome to Spartan Tutorials. Today we are going to talk about how we produce recombinant proteins in bacteria, E. coli and other prokaryotes. Okay, so various products, for example, antibiotics and many human proteins, for example, insulin, interferon, they are produced by cloning their genes into bacteria and using them as factories to produce these recombinant proteins. So how is it done? What are the factors needed to take into account whether we have good yields of these recombinant proteins? That's what we are going to discuss today. So let's get started. Now, as I said, microorganisms have been the source of many useful natural products. For example, right from the brewing of beer, since the prehistoric times, to the discoveries in the 1940s, starting with Alexander Fleming of the antibiotic, for example, penicillin, which saved thousands of lives in Second World War. Now we have ample number of antibiotics and other natural products which are isolated from bacteria routinely. So tetracycline, erythromycin, vancomycin, these are just some representatives. Similarly, we have many antifungal agents like amphotericin B, anti-cancer agents, for example, bleomycin. These are clastogenic agents. So these help in DNA double-stranded breaks and are used in chemotherapy. So lots of different products are obtained from bacteria. Here are some more examples: biofilm inhibitory agents, immunosuppressive agents. So these are small molecules produced by industrial microorganisms, but we are going to talk about production of recombinant proteins using prokaryotes. Now here are some good examples. For example, granulocyte colony stimulating factor. This is a small protein, as well as humulin, which is human insulin in the recombinant form, first produced by Eli Lilly, and it was granted a patent and approval in 1982 by FDA, one of the first recombinant DNA products to be commercialized. And here are some more examples: human insulin, interferon alpha, human interleukin 3, granulocyte colony stimulating factor, GCSF. It is used in chronic myeloid leukemia, human serum albumin, a very important blood protein. So there are a couple of different strategies to go about first cloning the gene, because it all starts with cloning the human gene or the relevant gene responsible for that protein. You can go with a cDNA library, so that starts with isolating mRNA. Here we are taking the example of a mouse gene, which may be similar to human gene, but we can also use human mRNA. We can reverse transcribe it to cDNA using reverse transcriptase and associated techniques. And we can add synthetic linkers for cloning, for restriction enzyme mediated cloning purposes, and that will give our desired gene of interest in a cloned form. Another strategy is to isolate genomic DNA, and if we know the sequence, which we know for many organisms now, especially for humans since 2001, we can PCR amplify the respective gene and then clone the gene. And the third strategy is Gibson assembly or oligo-mediated assembly, which is followed by PCR amplification of oligos. So this is in vitro synthesis of DNA. Here we have much more flexibility because we are not limited by the template design or the template sequence. We can make any DNA that we want using our own mutations. We can introduce our own DNA changes and then make the gene responsible, and these will all lead to production of our cloned gene, and this is our starting material. So traditionally, batch cultures were initially used to produce recombinant proteins, but nowadays mostly fed batch cultures or continuous cultures are used, where we continuously provide nutrients to the growing bacteria and we are siphoning off the products at regular amounts of time. This is the first paper in 1977 reporting the expression in E. coli of somatostatin. So this really started the revolution of recombinant DNA technology and production of recombinant proteins. The first product was, of course, human insulin that was produced. And here we have the bacteria. So this is the host, which contain a chromosomal copy of T7 RNA polymerase. So we want very high amounts of our gene to be produced. So usually T7 RNA polymerase is very helpful. So its promoter is already cloned in upstream of the gene that we have, and we use various vectors which we will see, which are used to produce this protein. So here is the E. coli promoter with its canonical -10 and -35 regions. Usually these are also optimized sometimes, and stronger versions are used if we need very high inducible levels of gene expression. So we can use either an inducible system or a repressible system. So there are a couple of differences between those. An inducible system relies on an inducer. For example, we can use LAC system, which will LAC operon. So LAC promoter depends on IPTG to dissociate LAC repressor from the gene, and so the gene is normally off. If we add regulatory chemical, in this case IPTG here, it will dissociate LAC repressor and the gene will be turned on. A repressible gene, on the other hand, is always on by default, but only when we add a regulatory chemical it is turned off. Okay, so they have their own advantages and disadvantages, whether you want to have a system which you can turn on by choice or turn off by choice. So depending on that, you can use different vector systems and different promoters to do that. So these are the fundamental things that we need in order to have a proper gene expression. We of course want to have the proper complete clone, complete open reading frame of the gene that we want to have. We need a promoter, just like the E. coli promoter that we had. We need a ribosome binding site. Okay, this is the Shine-Delgarno sequence in case of prokaryotes. And we need a transcriptional terminator. So these are the essential components of a protein expression vector. And various types of bacterial promoters are available for our use. For example, as I already talked about the LAC promoter, it will lead to transcription when we add IPTG. TRP promoter, this can be induced as well as repressed. It can be induced by 3-beta indole acrylic acid, and it can be repressed by using tryptophan. So very good on and off system. TAC promoter is a hybrid promoter which combines the elements of both the TRP promoter and the LAC promoter. It is still inducible by IPTG, okay, and it is stronger than both of them. We also can use lambda promoters, which are inactive at below 30 degrees Celsius, so there is no transcription here, and at above temperatures of 30 degrees Celsius the gene is induced. The T7 promoter is really one of the strongest, and it is the strongest out of all these choices available. So IPTG can be used along with that. So we can use LAC operator to induce T7 RNA polymerase, and then T7 RNA polymerase can bind to the promoter upstream of our gene to drive the expression of our protein. So this is the design of a protein expression vector, and here we have different components. So T7 promoter is there, okay. LAC operator site is there for binding of LAC repressor. Ribosome binding site is there. Here is our ORF in green, so that is our gene. T7 transcriptional terminator is there. Ampicillin resistance gene is there to select for transformants. Origin of replication of the plasmid PBR322 is there. ROP gene is there. Okay, this basically restricts the plasmid copy number, and LAC-I, which encodes LAC repressor, is there, so it will make the LAC repressor which will bind to the LAC operator. So this is a very simple system, and we are only talking about E. coli in today's video, and I just want to mention some problems that can arise in this protein expression system. Although it's a very convenient, very simple system, and it has been optimized so much that nowadays we can even have almost 50% of the cellular mass being composed of proteins, but still there can be some problems. And there are some limitations of the E. coli system. For example, there is no splicing, so it is good for expression of cDNAs but not for expression of genomic DNA fragments containing your gene, because there will be no splicing here. Another problem that can occur with E. coli gene expression is that sequences which resemble E. coli terminator They can be present in the foreign gene, human gene or other genes, which are not problems in their own systems, but when they are introduced into E. coli, they resemble the terminator, so it leads to premature transcription termination. So that can be problematic. And another huge problem with E. coli is codon bias. So codon bias refers to the fact that many of the codons, they are not equivalently used by different organisms. So as we know, genetic code is degenerate, so many of the codons, they are present for one amino acid. So sometimes one amino acid can be coded by 4 codons or 6 codons, but all of them are not utilized equivalently by a particular organism. And what happens sometimes is the human gene is optimized for human protein expression and inside the human cells, but E. coli uses different codons, and so they are basically not optimized for that. So scientists needed workarounds regarding that. So one way is to introduce mutations so that we can optimize the human gene for E. coli expression. Another thing is we can introduce genes for those rare tRNAs. So different strains are available. For example, there is the Rosetta strain of E. coli BL21, which is available. This is very commonly used. Another is E. coli BL21 codon plus. These are different strains available from the vendors, so we can use to get rid of codon bias or to minimize it at least. And finally, if all things go well, sometimes what happens is the protein has solubility or aggregation issues and it ends up in inclusion bodies. These are massive precipitates of the protein that we are trying to overexpress in big bacteria, and you can see in here in this case, this huge amount of inclusion body is formed. But sometimes it is still not very problematic because the protein sometimes is still active. So not always a problem. So inclusion bodies still allow the proteins to be produced even if they are sometimes toxic to the host cells or the host cell doesn't want to keep it in the cytoplasm. Generally they allow the high levels of protein expression to continue. They are just being sequestered or being placed in a separate corner of the cell, but they are still there, and they can be easily separated using centrifugation because of this mass of protein that has been prepared by the cell for you. You don't need to isolate it from cytoplasm. So inclusion bodies, although they are not preferred, but they are not always a problematic issue. The other issue is with post-translational modifications. So there has been a lot of studies showing that E. coli does not post-translationally modify correctly the proteins produced in E. coli. Okay, so there have been some workarounds regarding that. So for example, phosphorylation, usually it is very important for many proteins. So that has been somewhat taken care of by expressing human Jun N-terminal kinase 1 protein inside E. coli, which can do phosphorylation. And about glycosylation, we have a separate glycosylation system from Campylobacter jejuni, and that has been used and has been transferred to E. coli, although the pattern of glycosylation is somewhat different from what we actually observe in case of human proteins. So the protein may not be active as we thought it to be. Okay, so there may be some differences. But overall, I think E. coli and other prokaryotes, there are other prokaryotes that I didn't mention, for example, Bacillus subtilis, Lactococcus, Streptococcus. These are commonly used in industrial microbiology and in fermenter systems around the world in industries to produce recombinant proteins, on a very large scale. So this has been a very good boon to our, well-being and availability of medicines. So I hope you like this discussion of production of recombinant proteins in prokaryotes. I will return soon with production of recombinant proteins in eukaryotes. So stay tuned for that. Till then, take care and bye-bye. In the meantime, keep reading, keep learning, and keep growing. Take care.